He was PDF in Israel and then in US. So he's a well-known scientist in the area, and he is already well recognized by Bioscience Award, Rajiv Goel Young Scientist Award, Samantha Sekhar Award by Orissa Vigyan Academy, as well as he's fellow of all three academies, whether it's INSA, NASI, or ISC. So all three major bodies in India. He's a member of that. More over that, he's a nice person actually. Whenever we interact, he is always very helping. So whatever question you have, you may ask from him. So Debasisa,
yeah,
you are welcome.
yes, sure. Am I audible to you all?
Yes.
Yeah. Thank you, Dr. Raghava, for this kind words of introduction, and I am really delighted to get an opportunity to come and talk in this conference, which is very interesting. As you must have seen from the lineup of the speakers, it shows how this field has developed in this country. And the first talk I listened to Professor Samir Brahmachari, who has played his OSDD and all these efforts where Raghav played a very significant role, has really helped in encouraging a lot of young students to take up this field, and the field has really expanded. So thank you. What I'm going to talk today, after this fantastic talk by Urmila over a whole range of viruses and the role immunoinformatics play. So my talk is going to be computational approaches for design of protein-protein interaction inhibitor to find new antiviral, probably for SARS-CoV-2. So this is a very specific application. So what I would talk to you is explain you first what is protein-protein interaction inhibitor, why we are looking for this. Before we go to that, as you know, this slide would be common to all of you. So this is what it shows is the SARS-CoV-2 virus with the spike protein protruding out and all. Now it encounters a host cell, and there is a receptor here, cellular receptor, which is HSE2, human ACE2 receptor. Using this interaction, it is making into the entry into the cell. And obviously, if it is a virus, it uses our host cell's machinery to replicate itself, and there are many proteins involved. So the purpose of drug discovery has been, can we target either some of the viral proteins or some of the important interaction between the host protein and the viral proteins to develop some antiviral molecules or things like that. And instead of looking for new molecules, if you can look for repurposed molecule, that means this molecule has been tested for some other drug, then you can really accelerate the process. So I will not go into these details. So these are some of the targets which people have used: RDRP, the MPRO, or ACE2 spike, which I am going to talk about. So this is shown here, and either the crystal structure is available or a very reliable model can be built based on a homologous structure that makes the things easy. So now that idea, before I go into, since I'm going to talk of protein-protein interaction inhibitor, I would try to talk of all drugs are not protein-protein interaction inhibitor. You also target a single protein, like the well-known example in the context of SARS-CoV-2 could be RDRP, like your remdesivir is the example. So remdesivir actually is a prodrug, so it is modified, and you have this molecule called REM-ATP. And as you can see here, it looks very much like the ATP, but instead of this hydroxyl group here, it has a isonitrile or a cyanide type of group here. So these two molecules being very similar, it can go and bind at the site where ATP should bind, and that is the way it stops the virus replication. And it was developed for hepatitis C virus, subsequently tried in Ebola and things like that. And of course, there are those, in some cases in SARS-CoV, it has shown efficacy. In some cases, its efficacy is questionable. So there are efforts to develop better drugs and things like that. So this one. Apart from this, there are other examples of single protein targets drug could be people are trying to design inhibitor for this viral protease called MPRO. And there are many interesting papers have come on design of inhibitor. MPRO is a protease. Can you design inhibitors for this purpose? But when a paper like this is published, of course, they have done experiment, they show by binding affinity, KD values for this drug, and they solve a crystal structure and all this. But how did they come up with this new compound? So in that stage, there are a lot of computational screening goes out. Like they might have reported 1 or 2 compounds which are finally high efficacy compound, but they would have screened it from thousands of compound library to reach here. So what we are going to talk is that is where the computational methods of drug discovery is going to help. So now in this talk, what I'm going to tell you is instead of targeting the single protein, like here, like you have a protein, like let us say it was RDRP or MPRO, this had its native substrate. So now you are designing inhibitor which mimics the substrate. So if inhibitor can bind, the substrate cannot bind, and that is how you are blocking the process. In contrast to that, in a protein-protein interaction inhibitor, there is an interaction between protein A and B. This could be the S spike protein of the virus, which is using the RBD, the receptor binding domain, to interact with protein B, could be the HSE2 receptor on the cell surface. So now you want to design a molecule which is going to bind, let us say the RBD, and because of that SE2 cannot bind. And so now instead of it, so this is your target is a protein-protein interaction pair, and that is what I'm going to talk about. So there are some advantages as well as there are challenges, and these are the things listed here. So usually if you are targeting a single protein, it will be a deep binding cleft and it is always easy to design. But whereas if you come to a protein-protein interaction inhibitor, it could be a flat surface, could be hydrophobic and things like that. So here there are some this one limitations as well as advantages listed. But over the year, there has been efforts to get over these, and there are many protein-protein interaction inhibitors are being used in the anti-cancer therapy and all. So there is some, what I am going to talk is some of the work was also done by, similar work was done by the next speaker in some of his earlier work, this one. So what, so now coming to these things, how are you going? So we were initially interested in designing protein-protein interaction for Plasmodium falciparum. We were like this, and as Urmila said, when this SARS-CoV-2 came, a lot of protein, it is not only the repurposing the drugs, you want to repurposing your expertise. So if you have to expertise to do some computational analysis or design a PPI inhibitor, you repurpose it. That's why some students got interested. Okay, can we take up this problem of apply this to PPI inhibitor for this one? So now what I will talk of is essentially this work. We have done a machine learning based approach to design inhibitors which can probably block the interaction between RBD and HE2. And this is the work which I will explain. And in contrast to that, we have also tried certain Pharmacophore based methods on other this one. So I won't talk about this. I will mostly talk about the left part of this one. And now this slide, you don't need introduction. So typically when you do computational drug screening or things like that, you are asked the question, okay, why you are doing this? Because finally it has to be experimentally tested. I have already answered it, because even if you are going to do experimental testing, you cannot do experiment for all 800,000 compounds. Say all different types of computational methods are screening is reducing your search space by doing a variety of technique. If you can come up with 20, 30 or 50 molecules which can be experimentally tested, so that will be the ideal way to do experiment. Otherwise, it is impossible to do experiment with each and every compound which can be a potential drug. So now coming to this slide, what I am essentially telling you is this one, drug discovery introductory slide. So if you ask people what are the different methods of drug discovery, people would say that, oh yes, there is a structure-based method. There is where you talk of docking, you need a 3D structure of the protein, you need a 3D structure of the ligand, you calculate the binding energy and things like that, which comes under the structure-based method category. And in contrast to that, there are ligand-based methods where you may not have any idea on what is the target of your protein. You know that this molecule is giving you the desired bioactivity, inhibiting the growth of the, let us say, Plasmodium in the cell line level experiment. You don't know whether it is going and this drug is going and hitting a kinase or a protease or a protein-protein interaction. So that was called the ligand-based methods. So now, as in the last decades or more than a decade, so what has happened is this machine learning is playing a very important role in the drug discovery process. Like it is true that machine learning was also being earlier used both in the context of structure-based as well as ligand-based, but it is because now it is gaining prominence because the amount of data has increased, and with deep learning, the efficiency of these machine learning models have increased. That's why I am putting it as a machine learning method as a different this one. But strictly speaking, one may say that quite many of the machine learning method, if it is using a ligand-based parameter, it could come under ligand-based method, or it could go under structure-based method. So now coming to the ligand-based method, so I would, in the interest of time, I would skip this slide. You don't need to the. Whereas the major challenges in the structure-based methods has been what you need to. You may have a structure of a protein, the drug target. You know the structure of the ligand, but how to evaluate the binding energy of the ligand in the binding pocket of the protein has been a major challenge. So one is you need to have a confirmation generation method, like you can evaluate different binding poses of the molecule inside the binding pocket. You should be able to evaluate a large number of binding poses, and you should be able to calculate their binding affinity fairly reliably. Then only you can say, okay, this molecule binds with high affinity, not the other one. But calculation of this binding affinity using a atomistic scoring function has not been always easy, and that is why people look for other methods which can get over this problem. But this is a real challenge, and of course, and now that the machine learning is coming to be applied with structure prediction and with the success of AlphaFolds, I think the day is not far where machine learning will also help you to evaluate, to develop scoring function which can do better evaluation of the binding affinity. So now coming to this scoring function, there is some of the scoring functions could be physics based, some could be entirely empirical, some could be knowledge based potentials. That means you analyze a lot of crystal structure, like Urmila gave an example of some of it during her talk, and see what type of interactions are favored in the crystal structure, which atom types are preferring to be in contact with which other atom type, and those are called knowledge-based potential. And of course, now you have a machine learning-based descriptor. So whether you are going for a structure-based method or this one, so it is essentially, finally, it comes to how you frame your scoring function. In the machine learning, you have reframed your scoring function in a little different way in terms of the structure-based this one. So now our goal here was to, whether it is for a plasmodium, we were looking for PPI inhibitors, or from the for an antiviral molecule for SARS-CoV. The challenge becomes like for plasmodium, let us take the example of the malaria. So there are a whole lot of compounds like GSK compound library. They have released in public domain some 13,000 compounds which they know which is inhibiting the growth of plasmodium. And there is even a further smaller box, or what is called MMV box, this one, Medicines for Malaria Venture, that has a list of few hundred compounds which are known to inhibit this one. So the idea is, can drug discovery people use it to develop better molecules which can be more potent and things like that? But the challenge here was, you don't know what is the targets of these proteins. So the target of these proteins, some of the compounds could be kinase. That means it is targeting a single protein. It could be a protease, or it might be targeting a PPI inhibitor. So now our question was, can we come up with some sort of a computational method? Let us use machine learning for this, which can distinguish from these compound libraries which are the likely to be PPI inhibitors or not. And once you know which are the PPI inhibitor, we can probably go to the next stage and say which PPI it might be inhibiting. So I reframed the project challenge into two different small sub-bits of this one. So now, if you are going to develop a machine learning based method for identifying which are the potential PPI inhibitors, this one you should have, all machine learning requires a training data set, and that is where, using this data set, you will train your machine learning model. So you need some examples of which are the molecules which are known to be PPI inhibitor. And as Urmila talked during the talk, you also need a negative data set. For any of the machine learning problem, the choice of negative data set is equally important. You need set of molecules which are not known to be PPI inhibitor. So now we looked in the literature. So there were many databases where it is listed these are the different types of protein-protein interaction complexes which have been targeted by different methods. And so like MDM2P53, which has been targeted in the context of cancer, RAS, SOS, most of them would be in the context of cancer. There was not that much data available for RBDSE2. As when we started the work, we added some data in that direction. So by going to these databases, we could get information about known PPI inhibitors. So what you find is, you could find some 2,578 compounds which are known to be inhibiting a PPI in somewhere or other, and they are targeting 27 different PPI, and these are the databases from which this compound list was taken. And once you do that, so that is your 2578, which is your positive data set. So now you need to have a negative data set, and quite often in the developing this model, to have a negative data set is not easy, because somebody would have reported in those studies, okay, we tried this compound for this positive data for this PPI inhibitor, it didn't work as an inhibitor, but those data is not really compiled very systematically, so getting it from a database or thing is not easy. So you make some approximation about what would be a good set for the negative data set. So for this, we went to ChemEMBL, and wherever you have data about the compounds and what is their target. So this could be anything which is targeting an enzyme. It could be a kinase, it could be a protease or a ion channel or a receptor. So because we know these are single proteins which are being targeted, from that we pick the negative data set. And of course, you have a negative data set, you have a much bigger negative data set, much smaller positive data set. You have this imbalance in the positive and negative, that problem, but you can tackle it. There are different ways. I won't go into this. Let us say we randomly choose the equal positive and negative data set from it. And now for the machine learning, so what you would do is you would keep a portion of the data out completely, which is for entirely test data set. And for this data, remaining of the data, you will use as a training and a validation set, and using that you will train your model. So you could use two-fold, n-fold cross validation. I will not go into the details here. And you can use a variety of classifiers. We tried different, but what the result I will present is the random forest classifier. So the next thing here is, so in this case, for many of these, this one, we didn't have an idea how this PPI inhibitor exactly, even though we may have a structure of the PPI complex, but we don't know how this ligand is going and binding there, if at all it is. So in absence of that data, this machine learning model essentially became a ligand-based model. So you try to develop the model using only the ligand-based methods. So there are a number of different properties you can use as the feature vector. So this is the physicochemical property-based feature vector. So these are how many bonds are there, how many atoms are there, how many hydrogen bond donors, how many acceptor, how many aromatic ring. In terms of that, every compound will be represented as a vector, which all the physicochemical properties you can develop this one. And using this feature vector, you will train your model and test how good it is predicting. And of course, this I would skip in the interest of time. Most of the people who are attending this conference would know that when you evaluate this method, you will use a ROC curve and the precision recall curve. You will count many different statistical parameters, and which are essentially coming by counting how many true positive, how many true negative, false positive, false negative you are doing. And this is essentially a binary classifier. You give a compound, it is telling, is it a PPI inhibitor or not? And that happens to be the limitation depth, but you still need that to give in a compound you want to tell whether it is this. So under that context, when we train using the physicochemical property descriptors and a random forest model, so what you find is the best prediction you can do probably is giving you a AUC of 0.77 or 0.81, depending on whether you are doing a leave or out or a two-fold cross validation. And these are the precision and recall values. So now the question is, like, that means with this model, if you are working at a false positive rate of around 20%, you will only predict 60% of the true positive. If you want to go to a higher true positive, you would get more false positive. What would be your goal? Your goal would be to improve how you can get more true positive at the cost of less false positive. So this curve has to be more convex so that AUC value has to improve. So what can we do to do this model? So can we think of more complex feature vectors which represent properties which are just not just physicochemical? So in that context, we tried looking at it, and in chemoinformatics is highly enriched field. People have developed a lot of different types of fingerprint. So based on literature and some of our earlier work on metabolite chemical structure prediction, we zeroed it on this, what is called a Morgan circular fingerprint, or it will also be sometimes in the literature will be told ECF3 extended connectivity fingerprints or things like that. So what it is doing is, so instead of just calculating physical properties, in this fingerprint is using all types of different substructures which are present in your this one. I will explain it little more. So now by this, you say, like, you go to every atom, you draw different radius. This is with radius 0, that means atom itself. You draw a radius of 2, you just take the next neighbor, one bond this side, one bond other side. You go to the radius 4, you get 2 bonds this side, you got 2 bonds other side. For each of these different radius, you try to count which other atom types are coming, what type of bonds they are, single bond, double bond, or what are the properties of the atoms. And this way you build your fingerprint. So that means this has automatically the atom type as well as connectivity information gets built into your model. Whereas if you are in the earlier model, what I gave example, physicochemical property, the connectivity information is not coming very explicitly. And this was the advantage of using this model. And what you see here, the earlier, whatever same data set has been used, what you are getting a AUC of 2-fold, which was like a 0.81. Now it has improved very significantly. Your curve has become more convex. And now with just a 12% of false positive, which will be somewhere here, you are able to predict with a 84% of true positive. So by just choosing the right fingerprint, you have, right feature vector, you have improved the prediction accuracy of your model. And that is for the students, it is very important. That is a major challenge in developing new machine learning methods, because you have to understand your data. You have to understand, because sometimes people think to say machine learning is like a black box, but it is not really what is called the interpretable AI or things like that. These are the things you need to think of. So if you think of all these features, you will be able to develop better machine learning models. So I would, and of course, it was validated in terms of other statistical parameters like MCC, F1 score, this one. But and what we showed is it is not only the validation data set, even on the test data set, which was entirely kept out, you are able to do good predictions with AUC values which are much higher. And the other question is, like in this, the Priya was the student, he was doing this work, and she tried a lot of different things. And okay, we have used, let us say we are using SBM, like if you are using Weka package, it allows you to play around with random forest, naive Bayes, or any other types of machine learning training, or even deep learning. So nothing was significantly improving the performance over other, including deep learning. But of course, in deep learning, you would not have tried all the features. So what it says is, whatever result you have got, the type of machine learning training you are using is not making that much of a difference. And the other question which you have to answer is it picking compounds which are just around your scaffold, or it is picking compounds with a very diverse scaffold? So what is told in this TSNE plot is a clustering of the compound using the chemical feature vector similar to that feature vector. So what it is saying is, there are compound libraries which experimentalists use for screening, is a ChemDeep PPI library, let us say. So these are a lot of compounds which takes all types of this one. So now the training compounds are the red ones, and the pink compounds are the ones which our model is predicting. As you can see here, they are not just clustered around red, they are covering the more or less the entire chemical space. So that means the model, it is not just a overfitted model which is very close to what it was trained on. But the next question would be to, now given a compound, you are able to tell which, is it a PPI inhibitor or not, but you would be interested in getting machine predictor for RBDS-E2 or predictor for CD4-GP120 or BCL2 or things like that. How do you achieve the next stage? So of course, you need data for doing that task. So if you go to class-specific information, if you go to this data, you find that the data is really scarce. Like BCL2, you may have only 80 compounds which have this one. And after doing the clustering, we have done a clustering at 80%, like you don't want to include compounds which are very similar to each other. So now these are, so for all classes, it is really difficult to build model, but at least for the classes where there are 30 or more compounds are available, you will be able to build by using a similar model, the class specific predictors. So now what is shown here is, I will not go over the details. So these are the machine learning model like bromodomain, histone, BCL2, BAX, this one like IFA, ICAM or RAS SO1, these are the classes came. And that is the time when the SARS-CoV virus data was coming. There is an experimentalist group published a set of compounds by screening a large number. They had identified 30 compounds which are potentially inhibitor for RBD-HSE2. So of course, it was an opportunity for us to use this data and to develop a classifier for it. So of course, so that is what is shown in this class. And this really doesn't have that many positive data point, but I would go into more details into it. But of course, this classifier gives you an opportunity to look for potential molecules which can be used to inhibit the interaction between RBD and SE2. And now, so what the general predictor which you have developed, using that you can find out which are the molecules which can be a PPI inhibitor irrespective of the class. And in the next stage, you can predict whether it is likely to inhibit RBD SE2 or not. So we could get somewhere around 600 compounds which would fit this criteria based on this machine learning model, and they go to 300 different diverse chemical scaffold. So here and now, of course, where it has to be, this is where a challenge comes. Like this is a binary prediction. We do not have understanding of how it is interacting, how good is its binding affinity. Because of course, you can develop really quantitative regression models to predict, but your data should have that type. So this is where is the challenge. So that is where we switch over to the structure-based method. So once you get those compounds, you can use a docking or things like things like that. So using that, you can find out what is shown here is the RBD SE2 complex that is on the left. This is your RBD and this is the SE2. And this is the interface you are trying to inhibit. And these are some key residues which are forming hydrogen bonds, and through molecular dynamics or other simulation, we know that these interactions are really important. So what we are looking for is, can our compound replace these interactions? And what is shown here is, when you take just the RBD and dock these, they are indeed, these small molecules are interacting at that site, and some of the complex where SE2 was binding, those interaction is being replaced by interaction with the compound. And that is how this compound, this can work as a PPI inhibitor. And this is just an example of one scaffold. So in contrast to that, and this is where I'm trying to explain you what was the real challenge in making this these things in a structure-based method. Like this is the paper what I said, based on which we have taken this experimental data. At that time, it was only available in bioRxiv. Probably this might have been published subsequently. I should keep track of that journal and update my slide. So this is targeting SE2 RBD interaction from as a platform for COVID-19 therapeutic using an experimental assay called alpha ELISA. It is ELISA type of this one. So for a repurposing purpose. So they identified 25 inhibitors, and but they gave a large data set which do not work. So that can really work as your negative data set. So using that, what you see here, if you use a machine learning method, you can really predict well. That is the ROC curve. But now let us say you used docking. So what you can see here, the scores for your positive data, binders and non-binders in terms of docking score, they are almost overlapping. Because of this, you will, if you use docking score as a criteria for filtering compounds, you will not be, your ROC is going to be only 0.52. That is as good as random or even worse than random. And this is where what your machine learning models are trying to help. Okay, using machine learning model, you can predict certain thing, and then you can use that to test how it is interacting or between a set of compound, all of which are predicted by machine learning model to be PPI inhibitor. What will be the relative binding affinity that you can use by you predict using a structure-based method. And but that is where the real challenge is in this field, how we can develop, integrate the biophysical principle of structure and the concepts of machine learning and newer modern techniques and improve this drug discovery. That is the major challenge. And as of now, what I told you is all this purpose of this talk, this conference, is to how to make this. It is not only do your research, publish or something, how these tools are accessible for people. And this is what I summarized, like this our work to develop this PPI inhibitors. So now that all this work is the predictor which we made is available in the public domain. And this is where we have a general predictor which is first predicting if it is a PPI modulator or not. Once you predict which are PPI inhibitor, you can go to the next stage. You can predict all these different types of clinically important PPIs, and RBD-SE2 is one of them. And this one is this is all the source code and things are downloadable along with the program, and we also have it available on a web server, SMMPPI web server from our site, where one can just come and paste the SMILES code of a compound. One can check whether it is a PPI inhibitor or not. And it has also done little more, all the PPI databases which we gave. So using a web interface, you can visualize each and every experimental data which has gone into its training. So apart from Priya, Manish is one of my lab member who has contributed towards the development of the web interface or things like that. So this is as of now what I summarized is this is the work which is done. I will now, in remaining part of the talk, what is available, I would just try to touch on some other structure-based approach, how we can this thing. So now we have developed a predictor. It is predicting a RBD-SE2, but as you know, this is really on a small data set. So the experimental data has to improve. And the binding affinity prediction is the real challenge, because if you want to develop high affinity predictors, you have to have different methods. It can only give you a starting set of compounds for further study by using this machine learning classifier. And this is not only for RBD-SE2, any type of drug discovery, these are the challenge, like the ones which integrate machine learning along with structural feature or the binding affinity. Those methods are not that well developed. So these are some of the limitations of the model I talked about. Only ligand-based features are used. Interpretation of prediction is difficult in absence of molecular basis. Drug design is with desired chemical moieties, like when you are doing a binding, like if it's binding at this site, you found this interaction is hydrophobic, and the receptor, the residue is interacting with charge. So this methyl group, you may put a hydroxyl group. So that is where your interpretation easy and design was also easy. But whereas with a machine learning, that aspect becomes different. So that is where the structure-based and machine learning has to integrate better, how automatically the design aspect can come. And what I am suggesting is, of course, the structure-based features with advanced machine learning techniques can help in getting over some of these limitations. But there is one more in the remaining part of the talk. I would talk of one more limitations, like when all this data which we take, let us say our positive data for predicting RBD-SE2 inhibitor. So which RBD they would have used? Most likely it was in the beginning of January 20, the most likely the RBD they would use the RBD which was there in the original Wuhan sequence. And as you know, the virus is mutating. We have alpha, we have delta, we have all these things, and it played havoc. That means on this, so what is shown here, this is the spike protein, and it is a trimer, and there are 3 RBDs, and usually the one RBD is in the up conformation which is interacting with SE2, and 2 are down, and this is the RBD-SE2 complex, and this is the interaction we talked about that we inhibiting. But if there are some mutations, let us say you designed certain the inhibitor by this machine learning method. Now you design something else, some mutations. The mutations are constantly coming on the virus, the whole spike protein, many places, and many mutations are including on the RBD and the interaction interface. So the inhibitor we predicted may not work for the delta strain. So you need a model which takes into account all these, the sequence information, structure information of the receptor. And this is where the these atomistic or the structure-based methods have an advantage, because here, if you want to incorporate all that, as you saw in the previous talk of Urmila also, you need the complete structural information like antibody antigen complexes you can build. And this is for the students. If you want to build such models, you have to make a protein-protein interaction or a protein-drug model with all atomistic details, and you have to calculate interaction between the atoms using electrostatic, van der Waals, bond energy, dihedral energy, or things like that. But the challenge becomes, this becomes a a optimization problem locating the minima which corresponds to the either drug binding or the protein interaction binding on the free energy surface of the system. And this is where is it because of the multiple minima problem, locating the global minimum is a challenge. But there has been work, and if you know, 2013 Nobel Prize in Chemistry was given for molecular dynamic simulation, or these are the type of atomistic simulations people like Martin Karplus, Leavitt, and Arieh-Warcel developed. So these were all built on the work of Professor G.N. Ramachandran, which I showed you in your earlier slide. So now we can use the in some such techniques. So what we have tried doing it in my next few slides, I will just give you a very quick overview without going into the details of the work. I'll just tell you the some perspective. So the same problem of finding RBD-SE2 inhibitor we have tackled using a atomistic simulation method. So over period, what has happened is like many other people have designing. One is we were looking for small molecule inhibitor, but what people are looking for is, can you make peptides as inhibitors? That means you look at the interface, can you design a peptide which can block it? In that context, there are so many crystal structure has been available. Their binding affinity is known. So this is the whole RBD-SE2 complex. If you just take one helix of the SE2, what is the binding affinity? The whole complex had a binding affinity of 75 nanomolar. If you are taking just a single helix, the binding affinity is 1300 nanomolar. So if you take two helices, the binding affinity turns out to be 42 nanomolar. And these are the different strengths like alpha, beta, delta strengths. These are the binding affinity of SE also varies because the RBD is catching up some mutation. The interaction is changing. And same way, in the David Becker's group, they designed what is called mini proteins. Like these are not like they have used couple of helices and make proteins using Rosetta protein design software. And here they can say really go towards the picomolar inhibitors like 0.5 nanomolar, 1 nanomolar, 21 nanomolar. These are called mini peptide complexes. So for all of these, the crystal structures were available. So that gave us the opportunity to we can really use atomistic simulation methods, do molecular dynamic simulation, where you can compute the binding free energy using what is called MMGBSA, molecular mechanics, generalized bond solvent accessibility methods. So I will not go into the details of those methods, but using that, we can address the type of challenge I mentioned. But there is one more issue. So these simulations are very expensive. If you have access to a supercomputer complex or a machine with GPU cards, you can do it. So now in the spirit of public domain tools, like this is not really amenable to be available as a public domain tool. But what people are doing, one of my group members, Pawan, has been really enthusiastic. So what his idea was, like other people have done for the other context, okay, we have so much of the simulation data. So can we analyze this data and develop a pharmacophore model? So this is the way the pharmacophore models are developed based on analysis of crystal structure or single protein. Here is a pharmacophore model developed based on binding the molecular dynamics of the RBD-SE2 complex. So now all this data can be made available in the public domain, and if somebody comes with a new compound, you can use using the pharmacophore. So even if somebody doesn't have access to all the simulation, he can still build these features into the model and search it. So I will just quickly tell about our work in that direction. So this is where so what we did is, of course, we analyzed all these RBD-SE2 complexes. A variety of analysis was done, and of course there are challenges. You really do not reproduce the experimental binding affinity. In some cases you can match, some cases you cannot match. But instead of going into all these details, what I will summarize is, so what we did is the RBD surface can be put into these three different regions. You can say binding site 1, binding site 2, and the binding site 3. And in all these different complexes, the region they are binding is in binding site 1, which residue is going to interact. In binding site 2, which amino acid is going to interact. In binding site 3, which amino acid is going to interact. And because of that, the binding affinity is changing. So by analyzing these complexes, we have been able to get a matrix on each of these binding sites, which amino acids are preferred. So now using this model, it is easy to look for peptides or compounds, which you would say that, okay, I will design a peptide. So because of this, now I am putting the residues according to their contribution to the binding energy. I will be able to make a peptide which binds with higher affinity. So I started with one peptide. Let us say it had a delta G of minus 47 kilocalories per mole. And using this binding site matrix, which we have derived from this very long molecular dynamic simulation, I keep changing the residue. So now you can see here, if I made a T22M mutation, I got it minus 52. If I made a E37 to Y, I got minus 55, and things like that. So you have been able to change your binding affinity by almost 8 kilocalories by making one mutation. And this data, we are Himanshu and Pawan have been involved in this work. This we are really getting from the MD simulation and our MMPBSA analysis. So this is a way, by analyzing this MD trajectory, you can think of how you can build better inhibitors, or that in this case, this is the peptide-based inhibitor, but you could design the non-peptide-based inhibitor. And that is where I was going to talk about this work of building Pharmacophore based on dynamic ensemble of structures which are sampled in the MD simulation. And this is what is done. Like you did MD simulation, you got this, how this one, and based on you pick the set of conformations which are stable and contributing to very good binding energy. Now you are deciding them into pharmacophore features using which you can screen compounds like this. I don't have time to get into this work. This sort of colored spheres are going to be like, if this green is, so it could be a pitch, some molecule will fit here which can potentially form a hydrogen bond. This red could be hydrophobic. This could be have a both polar as well as non-polar feature. So this, depending on different types of chemical properties, these regions in the space are depicted. So this has some degree of three-dimensional information as well as the atom type information. And because we are getting from simulation, we have some idea with which residue this pharmacophore is going to interact and how it is contributing to the binding affinity. So using that, you can make your real dynamic pharmacophore model. So what in this slide we are showing is it different from a pharmacophore people have done from a static pharmacophore? You can take one RBDSE2 crystal structure complex and from that you can derive. So here what is it is showing the comparison. Features with green color labels are identified by both models, while circle features only identified by the dynamic model and things like that. So what you see is you find all the features you get from the static pharmacophore model, and you have got few additional features which comes from the dynamic pharmacophore model. So using that, and this is the sum of the final result what I was going to search. Now if you use this dynamic pharmacophore, I screen a library of ChemEMBL of 1.952 million compounds. Because it is a pharmacophore search, it is very fast. You can find out 188 molecules which can be potential PPI inhibitors. And you can even search for protease inhibitor, like this is not a protease, but still the protease inhibitor may have some structural features. Using that, it can mimic the binding interaction between RBD and SE2. That's why it is working as I said. You can find 31 protease inhibitors which have been tested in various, this is drug, this one, because there are no, so this is, you can find they could be potential RBD SE2 inhibitor. And we also searched, because in some of our work, we analyze genomes to predict the new natural products like RIPs or secondary metabolites. So we searched a natural product based library. From 185 compound, one can find 105 non-ribosomal peptide or cyclic peptides with cross links, which can become also potential RBD SE2 inhibitor. So these are some of the examples are given, like thiocylline, thiomuracin, or micrococin. These are the natural products which can also be potential inhibitor. So they have come from this dynamic pharmacophore search, and this dynamic pharmacophore is built based on molecular dynamic simulation for SE RBD2 complexes. So what I would do here, this is in the interest of time, I had to go very fast. So I would stop here. And let me acknowledge the National Institute of Immunology and Department of Biotechnology for the financial support under the BTIS program, which has been a very good program, which has like a lot of bioinformatics development in the country has been possible due to the BTIS program of DBT. And of course, we have got financial support from CDAC and National Supercomputing Mission, which is a program of the Ministry of Information Technology. So I would stop here and take questions, if any.